We're going to start the next session on aquarium development. Unfortunately, as we heard in the General Assembly the other day, Pascal Devanach will not be with us today, so I'll be chairing this session. My name's Rob Hicks, and I'm from Sea Life. I have a great group of speakers, and I'm very sorry about any of my pronunciation, but we will kick off, and we'll start with Michael Laterveer, who most of you already know. But just quickly, in case some of you haven't heard earlier, Michael started in Rotterdam to carry out some developments there and looking at some research just as the aquarium was starting to open. He's been working there now for 14 years and very recently this year took the post of curator. Looks like an interesting film. Michael will now do a quick presentation on cold water coral reefs, a new topic for aquarium exhibits to enhance public awareness of the threatened ecosystems. Thank you.
Thanks for the introduction. Sorry that you have to see my face again here. But I'm very glad that I got the opportunity to do a second talk we gave at the International Aquarium Congress about corals again. And I'd like to take the opportunity to thank Pablo and his staff for a really perfect congress, and I really enjoyed it. Do corals rock? Well, sometimes they do. It's not always easy to transport, and as Genoa showed for the transport of the cold water species from the Arctic, you could see here a boat of 70 meters rocking in wind force 11, and that was a coral transport. I will tell you more about it. Just a quick overview of what I'm going to talk about: cold water coral reefs, their threats, something about collection and transport, then exhibits and animal husbandry, and what can we do about the threats, exhibits, etc. First, cold water corals. Distribution. Not much is known yet about them, but the black dots on the map indicate where reefs have been discovered. Because they occur between 30 meters and 1500 meters depth, especially 800 to 1500 meters depth, have not been investigated very much yet. And regretfully, the first indications of coral reefs are because fishermen come up with some corals. And they especially occur around submarine banks and seamounts. They need some current to bring them food. And so elevated areas in the deep seas, you often find them. They can be very small, like 30 centimeters, or they can be vast complexes, like 30 meters high and literally kilometers long. A few reefs that are well known are the Rockall Trough and the Sea of Hebrides in Scotland, and near the USA, Florida, Cape Canaveral. In contrary to the warm water coral reefs, there are only a few frame building species. Lophelia and Madrepora are the two species that occur especially in the Atlantic, and the three other species occur mostly in the Indo-Pacific areas. But again, not much is known, and they occur from pole to pole. Here you can see underwater footage of a coral reef at, I think, at 100 meter depth, and it's a very rich area. You see they're totally white, pollux extended, and yes, they live in total darkness, and still they're reef building. So it's a very special habitat, and it's really the same as the warm water coral reefs. It's a very rich environment concerning biodiversity. Here you can see fish lying in the crevices, adult fish. This is a lang, I think, and a lot of food is also, because they occur along these seamounts, small food, plankton is coming nearby and gives a lot of food for the corals, but also for inhabitant species. Let's see if I get to the next slide, which is not easy. Maybe this one. That's working. Okay, take the other one. This is footage of the reefs, coral reefs near Cape Canaveral in 1980. You can see a lot of fish, gags, grouper-like fish. It's a very rich habitat, and please keep these pictures in mind. The threats to cold water coral reefs, Heather already said something about it. The climate change, acidification of the oceans. So the CO2 levels in the air will definitely have influence on the coral reefs in the oceans, and especially the deeper reefs will suffer a lot. Another threat is exploration of the seafloor, drillings, etc., and especially looking at the spoilage and making a lot of dirt underwater while doing it. But the most threatening at this point, except of the climate change, is deep sea fishing. A lot of the stocks of fish in the shallow waters under 200 meters are depleted in a lot of areas. So the bottom trawls and the fishermen are looking for new fishing grounds. You can see here the bottom trawl and their effect on coral reefs in the deep. If you do just on sandy bottoms, it's devastating already. But if you can see a coral reef, it's really doing a lot of damage. And here's the footage of exactly the same area in Cape Canaveral 20 years later in 2001. Nothing. No fish, no corals, just a few colonies alive. And it's a vast area that is totally destroyed. And who knows about these reefs? Even scientists only discovered them about 20 years ago. And so the politics and to take care of these reefs are way behind. Try to get to the next. That's it. So why should we work on cold water coral reefs? Of course, they're very difficult animals to keep. I think it's a very important, little-known habitat. In my opinion, the area of cold water reefs will extend is more than our warm water coral reefs. There will be more cold water coral reefs all over the world in comparison to the warm water coral reefs. They're heavily under threat, as I showed, mainly unprotected. But it's also very interested in an all-encompassing aquarium project. Oh, wrong thing. It's entertaining. It's a very rich, interesting habitat. The educations. The corals live off our shores. Even in the North Sea, these cold water corals occur. Whoever told that? So you surprise your public. It's deep and it's mysterious areas. So it's certainly you can do something with education. Conservation. There's again a very serious threat, but we can still act. We can do something about it. We can show our politicians, the fishermen, what they are doing. And not the least is the research. It's very hard to keep these animals, and if you can't keep the animals alive, researchers can do research, and we as aquarists can help there. So to contribute to knowledge, we contribute to saving maybe these reefs. Collecting and transport is of course very difficult. It's mostly open ocean, and how to get there. Well, there's one advantage. They're found as fouling organisms on oil platform legs. So that's maybe a way to collect them. And researchers are doing research on these reefs, and they collect a lot of material which is not directly used by them and literally thrown overboard. So it's a joint effort between an aquarium and either a research institute or like the British Antarctic Survey with the deep, and they went to oil rigs to look at the fouling organisms. And I myself went with the Royal Dutch Netherlands Institute for Sea Research. I got the opportunity. And what do partners provide? Something most of us cannot provide ourselves. Large ocean-going research vessels. ROVs. A lot of equipment and knowledge on how to use them and how to get there. Footage underwater. And access to places you normally cannot go. The vicinity of an oil rig platform is out of you cannot go there as a researcher or as a vessel, and you have to have permission. Researchers can get that, or other people can. So if you hook up with these guys, you can collect and you can transport. So the deep had a connection, and they made a special net to collect the corals growing on oil rig platforms. And the deep made a coral hotel, because these corals need to be transported. So collection sites where we worked. The oil rig was in the north of Scotland. That's this dot. And I was able to go on 2 different trips with the research vessel to the Hebrides, here up in Scotland, and to the south of Norway near Oslo. So these are the collection sites some of the corals came from. Again, it's not. So this is the research vessel of the Netherlands, the Pelagia. It's a 70-meter vessel, and you saw the beginning film that it was battling seas with 11 wind force. It's very well equipped. We have one, I'm very proud that the Netherlands has still a research vessel of this kind. And they use these containers as micro labs, but also as labs for visiting researchers. And we could put a lab like this and could put in things like we wanted to. So there's a lot of variation, small tanks. One of the ways the researchers collect is with the box core. And you can here see the size. In the middle is a cylinder, and on top of the cylinder you can see heavy weights, and they take care that the cylinder will drill itself inside the substrate which is dropped on. It's a scary talk I will give. Here you see the box core going overboard, and as you can see, the frame around this round cylinder, which is approximately half a square meter, is large. So if you drop this on a reef, it's not saving the reef really. It's really wrecking it. But it's the only way they get a piece of the reef in exactly the form it was down there. So they can collect until 1500 meters with the box core. And you see here that the box core has been closed on the bottom as well as on top. And then the research go to it. You have the same water which is just above the reef. They did a lot of research on that. And then in the box core you can see what kind of substrate you have sampled. It's not always clear if you're above a reef or not. They do very good senses and they try it. And of course they sample most not on the reef but around the reef. And there's special permission if you do that. So this is in the vicinity of reefs, and you see all kinds of animals, and you see also that it's rocky. Well, the researchers look at all the little animals, they collect them, and I was with the first trip, I could go as an observer, and if animals were left, I could take them to my cabin and take care of them. The first trip was very, very hard because they really didn't care about an aquarist being there, and they left buckets just on the side, and after 24 hours they said, oh I forgot, you can have it, although I was walking there. It was a bit frustrating, but it's also rewarding because the second trip it was totally different. Here you can see the box core sampling on a part of the coral reef. Actually, this was a sampling that there was no coral reef there, but still some corals. So this was, I think, a nice collection. Every box core is numbered, photographed, and an inventory is made. The corals are used by different scientists for different reasons. Here you see the yellow things here. They are sponges. They seem to be a threat for the cold water coral reefs. The sponges are growing more and more, and they're covering large part of the coral reefs. And we have a sponge expert on board who was looking at them and the other animals occurring. Scientist takes part of it. Here you can see the difference between an aquarist and a scientist. The scientist wants to do the same. He wants to keep the corals alive, but sometimes I don't understand that they don't look at the animals more closely in handling before they put them in an aquarium and then do their research. So that's also something that we can learn them, and and they can learn us, of course, also a lot. It's cold. It's very nice. This was one of the few big collections, and the scientists just need a few pieces. So you can collect large colonies with also the substratum underneath. Luckily there are other ways to collect. This is called a Van Veen grab, and this grab had a video on top, and you could follow the grab while it was going over the area where the reefs were, and at the spot where there was not just one colony and not too much to damage, they dropped it, and it was very good to collect just small samples like this, and get just one colony on top. Could also, once they tried to, they did a collection with the agassiz troll, just dropping it down and throwing it over the bottom for, they wanted to do it 25 meters, no longer, the minimum damage, and it was outside the coral reef area. Here you can see it was really full after only 50 meters on the bottom. Researchers don't care how they empty it, so kicking is okay. It's really scary to see that if you're there. And there was a lot collected, and there were no reefs on the images when we were on top, but there have been reefs there. You could see it, that it's old material. And there's a lot of stuff still living there. And, well, the researchers, it was lunchtime, so they drop it and they go have lunch. So I stayed there, sprayed it, and tried to keep the animals alive and get some animals out. On the side you see a box. This was also collected with a agassiz troll, and it's quite a nice big colony already. The first trip we had had a box, small tanks, separating the animals. A lot of animals you don't know, galateas, and if they will attack each other. So I separated sea stars, anemones, and a lot of small animals living there. Then could bring that to cushion stars, bring it back to the aquarium, and we had a 3 by 1 meter aquarium just for this first trip, and looking what we could do with the corals. And Dirk did with the students, we did grow experiments, and after this first trip we could show that we really were able to keep these corals and even let them regrow. We got some from the institute as well, and they had retracted tissue with bare columns, and while feeding them we were able to regrow them. So that's also why we thought, well, we must be able to keep these cold water corals. Besides coral colonies, you've got a lot of limestone, which is, I think, very important if you want to do this ecosystem. What I already said, very difficult is that you have very big colonies. And what you just saw were the small buckets, so I couldn't bring anything like this with me on the first trip. And the second trip, I was very fortunately that the Deep donated their coral transport system that they created for their exhibits. It's 2 tanks beside each other. One tank to keep the corals in, and the other tank with the filtration system. If you look on top, it's got a little sand filter, a protein skimmer, which actually doesn't work because the ship rocks a little bit and it doesn't help. A cooler, but the sand filter does a lot, and it was really helpful. And especially it was designed to be able to connect it to a ship, which is quite difficult because they use other electricity and channels there. And on the right side you can see the coral colonies like they were collected by the Deep. On the trip I went on, the second trip, I had the opportunity to take somebody else, so I invited somebody who had a mini ROV to make footage for underwater. And we were a little bit laughed about because they said, wow, this doesn't work on a ship like that. But we got creative, and we made a garage for this very little robot thing, like this, and with the cord, of course, towards the ship where we could see the footage, and dropped it in the water. And although there was strong current, this little robot was able to really swim against the currents and make nice footage. And now we have these footage in our aquarium as our educational exhibit. After the first trip, we knew, well, we can collect, and with the coral hotel from the Deep, we were able to get bigger colonies and also live rock. So we had an exhibit, and we changed it, put extra cooling on it, And as you can see it is a deep exhibit, approximately 5 by 3 meters depth. From the researchers, I got a lot of dead coral, which was just outside their research facility. So I collected that as bottom material. And then you have a real coral reef suddenly in your aquarium, which is amazing, because in these structures of coral reef you see everything, sea stars introduced, but you see all small crabs. A lot are white, you can hardly see them. It's full of all kinds of gritters, and I thought it was very important to have as much as possible the whole ecosystem there, especially because they collect it anyway. Well, these are a few of the gritters, feather stars, crabs of course.
2 minutes.
Thank you. Thanks to Monaco, and General also offered us we introduced a little bit later boarfish and snipefish, which are related to cold water coral reefs in the vicinity of the Mediterranean, not where we collect it, but they can belong to this system, so it's still a good ecosystem. But then we got uninvited beauties. Like half a year this aquarium was really running well. We had a very nice educational display with this video, and then these little gritters came up, growing a little bit white, flocks on the side, nothing to worry about. But then they started to grow over the corals, and it was a real pest. They grow over everything. So what could we do? You cannot take them away by hand or with brush, because you only multiply them. So we were thinking of biological control. So we got some nudibranch from Vancouver. The first one, the orange one, and they didn't really do their job, and they ate the soft corals we put in there and not the gorgonians, so that was a setback. But we have a few small snails, nudibranch in the Netherlands. We collected some, and they should eat only the hydroids, but we were too late to really counter the effect of the growth of the hydroids. So after 6 months it was perfect, and after 9 months the reef was really overgrown, and we couldn't really save these cold water corals. And that's I think that's the learning curve, that you first have to be able to either collect or culture these nudibranches before you start keeping larger exhibits of cold water coral reefs. For the coral reefs there's still hope. Cape Canaveral, now there's quite a big area which is protected, and the Darwin Mounds in the Rockall Trough, also a quite big area, is, well, relatively it's very small, but there are areas which really are protected now, and that's a very good thing. What we can do is raise public awareness. The Deep had their coral system in display. These are corals at the Deep, this Madrepora species, and they had big other displays. I really like to acknowledge now at the end the UNESCO, United Nations Environmental Programme. They have a report on the cold water corals, and with it goes a CD, and most footage I showed comes from this CD. The footage is taken, and it's great that these researchers do it, by Jan Helge Fossa of the Norwegian Institute of Marine Science, and John Reed of the Harbour Bridge Oceanic Institute, which has this film footage of this reef in 1980 and 2001, which I think very impressive and tells a bad story, but it has to be told. I'd like to thank Matthijs de Lange of the Sea Foundation with the underwater robot, the mini one, and Living Elements who work with the Deep. And last but not least, I'd like to thank the researcher who invited me on her ship, Connie Meyer, that she had the view that she invited a public aquarium, and we did a lot together, and it paid off. So are there any questions maybe?
Thank you, Michael. Does anybody have any questions?
Laura Castellano, Geneva Aquarium. My question is, do you think they need the pressure to survive, the corals? Because I was involved in a discussion about these animals. Involved the scientists where they have a big concern about the needing of this coral of pressure. So I just want to know what you think about it.
Yeah, thank you for your question. It's a much asked question whether animals coming from deeper regions of the oceans need this pressure to survive. I had a lot of discussions on board with different scientists. In general, until 1500 meters, animals don't need the pressure to survive. So you can bring animals from 1500 meters depth, not like boom up, but slowly. If you do that correctly, they can just live under atmospheric pressures like we are sitting now. Below 1500 meters depth, there are animals which need these pressures and even physiological adaptations that they really need this pressure. Not all of them. The giant isopods are shown in Japan, and they can handle it. So it's more the question if can you keep the water cold while bringing the animals up, and can you keep the surroundings where the animals live stable at the point that you transport them, and that's the most important thing. After that, food is important, but the pressure is not a real issue, in my opinion.
I think so too, but
Okay.
I wanted to know what you think.
Yeah.
Max Jansen, Burger Zoo. The hydroid problem you had, are they hydroids that came with the corals, or are they hydroids that are from the Artemia or from the aquarium itself?
I think both, to be honest. The system we use is a flow-through. It's a closed system in total in Rotterdam. Everything is a closed system, but it's a flow-through system with our North Sea exhibits. So our North Sea exhibit with many animals is in there, and also our herring tank is on the same system. The only thing we do is take care that this system is colder and suitable for the corals. But there's really a lot on it, and also hybrids. Probably they're eaten on the natural coral reef, and so they live in crevices and you don't see them. I've been looking closely, but I haven't discovered them. We don't have the problem in our North Sea or the herring tank anywhere. So it's mostly related to the food. Of course, the fine food, Artemia, rotifers, maybe some algae. But I think they're carnivores, so rotifers and rotifers. That as soon as there's more higher quantities, they will start growing. And we were really surprised because there were just a few patches, and suddenly bang. And we didn't go in and clean them, because then you break up these colonies, and every single animal will settle somewhere and grow again. But it's that's the challenge. We have the same challenge with our deep reef, where Aptasia, the glass anemone, is exactly the same problem, and there you can do the peppermint shrimp or another nudibranch, Eleodella. You really need them to have a deep reef exhibit to counter the effect of the growth of Aptasia. And I view this the same with the hydroids for the cold water coral reefs. It's just something we have to learn. And so we have to breed nudibranch to counter the... But I think they're both coming with our colonies as well as they will be in the system.
A couple comments. One is on the question over there about the pressure-sensitive enzymes. I agree. From bringing up fishes up on the Johnson Sea Link, when we were out, I was out on the Harbor Branch boats too at one point when I was in New York, and we were bringing up fishes, and we found that it's about the same depth of what you're referring to in regards to 1,200 meters or so, that you start having issues below that because of pressure-sensitive enzymes. Another comment about the nudibranchs is that Tequina generally is pretty specific on what it feeds on. It feeds on Jersemia and that, so it may not be the appropriate type of nudibranch to be putting on those other hydroids. A lot of those nudibranchs are very specific on what they feed on, so it's picking the right one. The Hermesenda that you had on there is more of a generalist, so that might work okay. And then just one other comment is that there's some deep sea corals off of New Jersey, New York called Strangia, and it's kind of interesting because it doesn't have zooxanthellae at depth, but if you go farther south in shallow water, it has zooxanthellae, so it's kind of an interesting coral that way.
Thank you for your comments.
Thanks very much. One last quick question, or we'll move on.
It's Pablo Montoto from Madrid. I was wondering, what about the visitors' reaction to this tank? When they come from a probably tropical exhibit and they see a colorful fantasy display from in the Pacific, and they reach this tank full, not so colorful, but which has so much work beside. How do we? You think this is a success, or?
That's another nice question. I had big debates within our zoo on that. It's very important. My job as a curator, and most of you, is to collect the animals and show them. And in my opinion, it's very important that the educational department takes part in what you want to tell. Because first, it looks very uninteresting, but if you tell people it's deep and nobody sees this, and you're very, it's a very special thing you can see here, everybody looks. Our cold water coral reef is situated in the North Sea, so there's no competition with the warmer coral reefs which come later in our aquarium. And it's a corner where also jellyfish are shown. And I think there's now a very nice educational display with two long signs which are actually attracting the people. Before, the signs were put in pretty late, after 6 months, but then the corals died off slowly. So it was really disappointing. But after they put in the signs and had somebody looking at that, what the effect was of putting signs and where people were looking at in our aquarium, and there was a major difference. People tended to walk by, and by accident they went to the jellyfish. It's a little corner. But when these boards were, these signs were placed there, almost everybody looked at the site, and our jellyfish display was first not that interesting. And now it's one of the most highly regarded displays. It's a very small thing, and it's opposite the cold water corals, and everybody looks at the cold water corals as well. I haven't done this research, talking to people in front of it, and what do you think of it. You need the boarfish and the snipefish to have something swimming there. And there's a few, it's always funny to see the children, because they see it. Oh, Mom, it's a little crab over there. And then but the adults, if you only have adults, they say, well, fish, go. So it's different. And I think you really need the educational display to pinpoint, wow, this is special. This is not something you can see every day. And then people start to look better. And then also adults see the little crabs or the special things. So thank you for the question.
Thank you very much, Michael.